Hello everyone and welcome to my channel.
Transport chain, that is ETC. So this occurs in the inner mitochondrial membrane and it utilizes NADH and FADH2. It basically extracts the electrons from these and then it transfers it to oxygen and helps to generate ATP in the process. So this is an aerobic process. In ETC, we have complexes 1 to 4 which basically helps to catalyze the redox reaction. Here, a process called coupling occurs, whereas in oxidative phosphorylation, electrochemical gradient occurs, is formed, which is basically of protons. And what does it do is that it helps to produce ATP. So one glucose molecule, it helps to generate 32 ATPs or 30 ATP. 30 net ATPs. So if we are using malate aspartate shuttle, then 32 ATPs. In glycerol 3 phosphate shuttle, 30 ATPs are generated. Malate aspartate occurs in heart and liver, whereas glycerol 3 phosphate occurs in muscle. Starting with NADH. So basically this NADH is produced in the mitochondrial matrix via malate aspartate shuttle or the glycerol 3 phosphate shuttle. So during this process NADH is generated. The FADH2, it is generated by the succinate dehydrogenase, which is a part of TCA cycle and forms the complex 2 in ETC. The protein complexes, these are resides in the inner mitochondrial membrane, and electrons, they participate in redox reaction and transfers to electron to transfer of electron occurs to oxygen. Now talking about the complexes, so there are total of 5 complexes, four primarily the ETC and fifth one is the ATP synthase complex. The complex one is called NADH dehydrogenase. So here 2 protons and 2 electrons are transferred to coenzyme Q. It also pumps 4 protons into the intermembranous space of the mitochondria. The second one is SDH, that is succinate dehydrogenase, which pumps 2 protons and 2 electrons to coenzyme Q. It does not pump any proton into the intermembranous space. The first complex utilizes NADH2 and second utilizes FADH2. The third complex is coenzyme Q cytochrome C reductase complex. This helps to set 2 protons from coenzyme Q to cytochrome C. Along with this, 4 protons are pumped into the intermembranous space. Then complex 4 is cytochrome C oxidase. So oxygen is converted into water via cytochrome AA3 complex, and 2 hydrogen ions or protons are pumped into the intermembranous space. So in this there occurs transfer of electrons and it helps to generate a gradient which supplies or powers the ATP synthase, which is our complex 5. In this process, 4 protons are pumped back into the mitochondrial matrix through this ATP synthase complex. Here, one molecule of ATP helps to form one ATP, and during this process of sending back the protons into the intermitochondrial matrix. The last phase is oxidative phosphorylation. In this, this occurs via the complex 5, which is also called ATP synthase. Here, four protons they are pumped to generate one molecule of ATP. So, what happens is we have complex 1. So here, four protons are pumped into the intermembranous space, and during this process, we have FMN and FES, via which the protons move, and uh, NADH forms NAD plus protons, and those two electrons are transferred to FMN and FES, and then ultimately to a complex 3 cytochrome. Now in complex 2 we have FED and 2 electrons are generated. So again here also these will be carried forward to 2 electrons, and 2 protons will be carried forward along with 2 electrons and protons from complex 1. These are all transferred to cytochrome Q complex. Okay, so while this process occurs in complex 2, you need to remember that no protons are pumped. But here via the cytochrome Q, 2 protons are pumped into the intermembranous space. Then comes the complex 3, which is cytochrome C reductase, right? So in this, the 2 electrons are transferred to ferrous sulfide. So it is and 2 protons are pumped into the intermembranous space. From FES, the 2 electrons are transferred to cytochrome C, which will further transfer it to copper, heme E, and E3 complexes, which is part of cytochrome C oxidase. And along with this, half of oxygen molecule plus 2 proton ions. So, 2 protons, they will transfer the electrons to water, and these 2 protons are again pumped to the intermembranous space. Complex 5 is ATP synthase, where all those hydrogen ions, the protons which are present in the intermembranous, will go into the mitochondrial matrix, and via during this process one molecule of ADP, it will form ATP. So this is the diagrammatic view of the complexes. Now inhibitors. So complex 1 inhibitor is rotenone. The complex 3 inhibitor, that is cytochrome Q C reductase, is actimycin. Then cyanide azides and carbon monoxide will inhibit cytochrome C. Oligomycin inhibits complex 5. Talking about uncoupling agents, so these will dissociate the ETC and oxidative phosphorylation process. Here, ATPs are not generated and just mere heat production occurs. So this occurs in cases of salicylic acid, 2,4-dinitrophenol, and thermogenin, which is our brown fat which helps to produce heat. Next is prolonged hypoxia. So what will do is that there won't be any oxygen. So there won't be oxygen to accept electrons. ETC won't happen, and ATP process doesn't occur.
Thank you very much for listening. Let's keep on learning together.